I'm Samantha.
Hi, I'm Sandeep.
And today I will show you a technique for making polyacrylamide gels with fluorescent microspheres inside that will be used to track cellular motion.
And later I'll be showing you how to track the displacements produced by the cells, and later we'll calculate the traction forces produced by the cells drawn on the polyacrylamide gels.
Before we begin the experiment, I will first give an overview of the technique that we will use to make the polyacrylamide gel with fluorescent microspheres inside. The stiffness of the polyacrylamide gel is determined by the concentration of acrylamide relative to bisacrylamide. The higher the amount of acrylamide relative to the crosslinker, bisacrylamide, the higher the elastic modulus of the gel will be. This is shown in the graph here. Now I will explain how we will determine the appropriate volume of fluorescent particles to add to the gel in order to achieve a desired particle spacing. Before we do this, we must know how many particles per volume are in the colloid solution that we already purchased from a company. We will use the following parameters that we know: the total mass of the solid, the volume of the colloid solution, the mass of each individual particle, and the volume of a single particle. We will use each of these parameters to derive an equation that will generate the concentration of particles in the colloid solution. The number of particles per volume can be represented by the ratio of the total mass of the solid in the colloid solution to the total volume of the colloid solution, divided by the mass of each individual particle, m bar. We also know that the mass of each particle is given by the volume of each particle multiplied by the density of the colloid solution. We will let the concentration of the solid in the colloid solution be given by C, and its value in this calculation will be 0.02 grams per milliliter, as given by the company. We will consider a solution containing particles that are 1 micrometer in diameter for simplicity of this derivation. Substituting the given values for our ratio of particles to volume, we get 0.012 times 10 to the 12th over pi times rho. To check our solution, we perform a unit analysis to show the units resulting in number per milliliter, which is what we set out to achieve. Rearranging our solution results in a general formula for determining the number of particles per milliliter in a colloid solution, where phi represents the diameter of the particle in micrometers. In our experimental demonstration today, we will use fluorescent microspheres that are 1 micrometer in diameter. The colloid solution comprised of these microspheres has a density of 1.05 and a solid concentration of 0.02 grams per milliliter, as provided by the industrial supplier. Utilizing the formula we just derived and the given parameters, our colloid solution contains 0.036 microspheres per cubic micrometer. In order to determine what volume of colloid solution to add to our polyacrylamide gel, we must first determine the dilution factor, beta. We can determine how much colloid solution to add based on how far apart we want each microsphere particle to be in our gel. S will represent the desired distance between particles. For our experiment, we want each microsphere to be 10 micrometers away from the nearest neighboring microsphere in any direction. This is represented by 1 microsphere per 1,000 cubic micrometers, and our S value will be 1 over 1,000. Using our calculated value of 0.036 microspheres per cubic micrometer in the colloid solution, we will need to dilute the solution about 50 times in order to achieve a particle-to-particle spacing of 10 micrometers in the polyacrylamide gel. Finally, to determine what volume of colloid solution to add to the polyacrylamide gel, we calculate 1/50th of the total gel volume. The total volume of one batch of polyacrylamide gel is approximately 5 milliliters, so we must add 100 microliters of the colloid solution to ensure our desired particle-to-particle spacing.
In order to make the polyacrylamide solution, we will first start with a sterile glass cover slip, which has been heated on a hot plate at about 160 degrees Celsius for 15 minutes. Next, we will add a solution of 3-aminopropo trimethoxysilane on top of the glass cover slip. We will allow the solution to remain on the glass cover slip for about 7 minutes, after which we will rinse it off thoroughly with DI water and then dry it for about 7 minutes on a hot plate. Once the glass cover slip is dried, we will add 0.5% of glutaraldehyde solution on the cover slip. This is the stock solution, but we will use an already pre-prepared 0.5% glutaraldehyde solution to activate the glass cover slip. We will allow the glutaraldehyde solution to remain on the glass cover slip for about 30 minutes. After 30 minutes, we will rinse it off thoroughly with DI water and dry it on a hot plate for about 15 minutes. Now the glass cover slip is activated. We are ready to make the gel solution. The chemicals required to make the gel solution are as follows: heaps, acrylamide solution, bisacrylamide solution, TIMED, ammonium persulfate, and finally the microbead solution. We wrap the microbead solution in a foil because it's photosensitive. Once all the chemicals are mixed in the required quantities, the gel solution looks like this. Now we will add the microbead solution to the gel solution. Make sure you shake the mixture well in order to ensure uniform dispersion of the beads. We will pipette out the required quantity of the mixture on the glass cover slip. Next, we will add a round glass cover slip on top of the gel to make sure the surface is planar. This also helps in controlling the thickness of the gel. We will now hang the glass cover slip upside down to make sure that all the microparticles come to the surface of the gel due to gravity. The gel polymerizes in about 30 to 40 minutes time. After the gel polymerizes, we are now ready to remove the top cover slip. Now we will transfer the gel to a petri dish. We will add some heap solution to the gel to keep it moist and hydrated until ready for use. Before plating cells on the gel surface, we need to first conjugate fibronectin on the gel surface. In order to do this, we need to first activate the gel surface with hydrazine hydrate solution. We have already activated the gel surface with hydrazine hydrate. We then wash the gel with acetic acid solution and twice using PBS. Now we are ready to plate fibronectin on the gel. We will first aspirate all the heaps from the petri dish. We will add a pre-prepared solution of 50 micrograms per ml fibronectin on the gel solution. We will leave the fibronectin on the gel surface for about 40 to 60 minutes. After 60 minutes, we will rinse the gel surface thoroughly with some PBS. Next, we are ready to plate cells. We will show you the procedure to detach cells from a culture flask and plate on the gel surface. First, we will aspirate all the cell media from the T75 culture flask. Next, we will rinse the cells with some PBS. Next, we will aspirate all the PBS from the culture flask. In order to detach the cells, we will add about 5 ml of 0.05% trypsin. We will incubate the cells in trypsin at 37 degrees Celsius for about 15 minutes. After 15 minutes, we'll add about 5 ml of cell media in order to neutralize the cells. Next, we will transfer all the contents of the T75 flask to a 15 ml centrifuge tube. We will centrifuge the cells at 1500 rpm for 5 minutes. After 5 minutes, all the cells will be deposited at the bottom of the centrifuge tube. Next, we will slowly and carefully aspirate all the media on top of the cells. Now, we will resuspend the cells in fresh culture media. Mix the media well using a 1 ml pipette to ensure that all the cells are uniformly suspended within the media. Now the cells are ready to be plated on the gel surface. Aspirate all the heaps from the petri dish containing the gel. Next, we will add the cells on the gel surface. We need very low density of cells on the gel. Hence, we will just use about 200 microliters of the cells on the gels. We will flood the entire petri dish with fresh culture media. Now, we will leave the cells in a cell incubator undisturbed at 37 degrees Celsius and 5% CO2 for about 12 hours. Once the cells have stabilized for about 12 hours in the incubator, we are now ready to start the experiment. We will first look for an isolated cell which is quite far apart from its neighbors. Once we find such a cell, we'll image the beads underneath the cell under UV light. This will give us the bead image under full force condition. Next, we will dissolve the cytoskeleton of the cells using SDS solution. This will ensure that all the cells are removed and the beads are relaxed to their original position. Now, we are ready to image the beads under zero force condition. This gives us the two images that we required for TFM. Now that we have learned the procedures to fabricate the polyacrylamide gels with fluorescent microbeads and obtain the bead displacement images, we are now ready to compute the cellular traction forces. This method of computing traction forces from bead displacement images in general is referred to as traction force microscopy, or TFM. TFM primarily involves three major steps. First is the fabrication of soft polymeric substrates like PA gels with fluorescent microparticles. This is followed by cell culture and imaging of the bead displacements. The final step is the post-processing of the obtained images to compute the traction forces. The displacement field is computed via digital image correlation of the bead images. The force field is computed through finite element analysis, and finally the force field is optimized to get rid of any pseudo-forces. Cells apply forces on the underlying substrates. The amount of force applied depends on the substrate stiffness among many other factors. If the substrate is made sufficiently soft, the force applied by the cells may be easily calculated by measuring the deformation. This is the underlying concept of traction force microscopy. The bead displacement gives us sufficient information about the deformation of the substrate. This information, coupled with the material properties of the gel, allows us to back calculate the forces causing the deformation of the gel. Here we can see some typical phase contrast images of monkey kidney fibroblast or MKF cells cultured on polyacrylamide gels. The image on the left shows 2 MKF cells 12 hours after plating, while the image on the right shows 2 MKF cells after 24 hours of cell plating. Notice that the cell spreading area increases with time. Next, we will compute the traction forces generated by the cells on the left. The reference and displaced bead images obtained after the SDS treatment are shown here. The bead images are compared to displacements by searching a matched area with multiple pixel points, such as 30 by 30 pixels. This area, usually called a subset, has a unique light intensity distribution called gray level inside the subset itself. It is assumed that this light intensity distribution does not change during deformation. The displacement of the subset on the image before deformation is found in the image after deformation by searching the area of same light intensity distribution with the subset. Once the location of this subset in the deformed image is found, the displacement of this subset can be determined. Several functions exist to match the subset from one image to another. Here we can see the displacement field obtained after digital image correlation of the bead images. Our next step is to create a finite element model of the PAGL substrate. Finite element analysis, or FEA, is a numerical method of solving engineering problems. FEA may be applied in structural, heat transfer, and fluid flow areas. FEA is extensively used in problems where analytical solutions are not easily obtained, or problems where mathematical expressions required for solution is not simple because of complex geometries, loadings, or material properties. In FEA, we replace continuous geometry with a set of objects with a finite number of degrees of freedom. The body is divided into a finite number of simpler units known as elements. A set of elements is referred to as a mesh. A problem is defined in terms of loads, stiffness, and displacements and is solved by setting up a large number of simple algebraic equations at nodal points for each element and combining for the entire mesh. We make use of a commercial FEA software called ANSYS. The figure on the left shows a meshed model of the PA jaw, while the one on the right shows displacements obtained from DIC applied to the respective nodes. Finally, we solve the problem in ANSYS and the obtained force field is shown here. Notice that there are pseudo-forces present outside the cell. Pseudo-forces may be generated during analysis due to various different reasons pertaining to bead distribution, imaging errors, DIC approximations, etc. In order to get rid of the pseudo-forces, we optimize the obtained force field using an iterative process. We assign the forces outside the cell as 0 and recalculate the new displacement field. We compare this with the original displacement field and arrive at a more optimum displacement field. This new displacement field is again fed into the FEA model to obtain a slightly optimized force field. We repeat this process several times to arrive at the most optimum force field. The whole optimization process is automated using a MATLAB program developed by us. Here we can see the optimized displacement field. And finally, we arrive at the optimized force field. Notice that the forces outside the cells are 0.